2. And so we left off on synovial joints and the components of synovial joints. And recall that all of these are structures that you find in all synovial joints, but they're just going to be in a different conformation or in a different arrangement in order to serve whatever synovial joint needs to be made. And so at this point, the labs are the same. They know that the mandible connects to the skull. And so even where the mandible meets the skull at the mandibular condyle, that is going to form a synovial joint with the skull, the temporal bone. So even up here, your jaw itself is a synovial joint. It has all of these properties, but it's just going to be really, really, really tiny by comparison. And with that, as we look at this next segment, a lot of these synovial joints will have extra structures. Those extra structures are accessory structures. These are going to be things that support the synovial joint and may or may not be there in the same amounts in just different configurations, but they're not everywhere, or not all the synovial joints have it, but it's just meant to be that you might find some of these structures within synovial joints. So that's why they're called accessory structures on this set of slides. And so something to address after that synovial joint is when you look at the overall ligament names, the names or ligament is going to mean something that connects bone to bone. If you look very closely, you can see one ligament reaching onto that bone and then connecting all the way at the very bottom. That's a ligament made out of what connective tissue? Does anybody remember? Dense regular connective tissue. And so dense regular connective tissue is going to connect bone to bone. All right? And tendon was the same thing, but instead of it being connecting bone to bone, you can see it's connecting a muscle to the actual bone itself. And so this is referred to as a tendon because it's muscle to bone. Sorry, let me rearrange my thing here. Here we have muscle to bone. And that's what a tendon does. And a ligament is the same thing, but it's bone to bone. That's the big difference between a tendon and a ligament. Yeah, sorry, there's a mosquito in here. I don't want to get bit. And so now, as we move on, there are little things that cushion that synovial joint, not just that downward compression. The synovial fluid, we said it lubricates between the bones, and it also adds a layer of compression, shock absorption. And so that means that when it goes down, it's going to completely be supported by the fluid inside. It's going to help increase or give it strength, downward compression strength. But also, there are going to be tiny little containers of a tissue, and they're called bursa. And these little sacs, all they have inside of it is synovial fluid. So aside from having synovial fluid within the synovial joint, you actually have these little accessory, tiny membranes, these little bags that contain synovial joints that are helping cushioning any movement or that might be occurring within that bone. And they're doing the same thing that synovial fluid does: cushion and protect, lubricate, and just keeping things apart from each other so they're not wear and tearing away. Fat pads are exactly what you think they are. It's adipose tissue. You can see that fat does mostly sit underneath the skin in that hypodermis region. And then eventually you'll find muscle, and you'd have to move all that muscle to finally see all the bones that we're studying. Even as you're looking at this, you're seeing really all the complication that exists really when you're looking at limbs. There's not much more than the skin that you learned about, the integument system, epidermis, dermis. And then at that hypodermis, you're seeing all the fat that exists within that hypodermis. And then around that fat and embedded in that fat, you have the muscles that are going to be connected to the bones through their tendons. And then finally, you made it to the bone that we've been talking about. What's the outer line, this layer called of bone? Periosteum. And then what is this region here, the tougher part? Only on the outside? Sorry, I missed right there. Compact bone. And so this region outside is compact bone. What do we call the spongy looking one? Spongy bone. And what do we call the cavity in the middle? Medullary cavity. What sits in here? Yellow marrow. What sits in this epiphysis area? What kind of marrow? And what is red marrow for? red blood cell formation or blood cell formation. And so they all, long bones, typically have that same layout. And so you're sort of seeing it all come together now with all the different tissues. And today we finish with skeletal muscles. We're really going to see how this works. And we're finishing up our joint accessories now. And then meniscus. Now this word can be confusing, and meniscus is made out of fibrocartilage. And what does fibrocartilage serve in the intervertebral disc? What's its function? Cushioning. And so you can see that there's going to be these little pillowcases called menisci for plural, but meniscus, you're seeing one here, you're seeing another one here. I'm going to show you better pictures of this in a second. But they serve as pillow pads for the bone above, and that's what a meniscus is. This is a common thing to hear. People say, oh, I have to get my meniscus operated on. And sometimes it's pinching or got loose, and it could be a wide range of issues. Next, you have the patella. We're going to see next week there's going to be the kneecap bone in the patellar region called the patella bone. And so that patella bone has a ligament called the patellar ligament. And then next you have the cruciating ligament. As far as you're concerned, this word cruciate means crossing. Crossing. And they're ligaments that cross the actual joint itself. And so, for our bursa, same thing as the synovial capsule does. Our fat pads, we know what fat does. It insulates, it protects. Keep it simple for this one. The meniscus is going to be that pad of fibrocartilage that protects. I don't have that on there. And we're just saying between opposing bones. Prevent wear and tear on the bones. And then now, these ligaments now that I mentioned, we have patellar ligament and cruciate ligament, and something I'm not very fond of the way the notes put it here. So you'll see on this side I separated a little bit to something that makes more sense. Here you have extracapsular ligaments and intra. And we know what extra means, outside, intra, inside. So outside the capsule, inside the capsule. If they're outside the capsule, then they're going to be called extracapsular ligaments. If they pass inside of that joint, through the capsule, it's called typically an intracapsular ligament, and the only examples you're going to have to learn today is cruciate ligament. And patellar ligament, you will have to have an idea. And something I want you to pay attention to, what's the difference between, if we just pay attention to the patella, and we look at the proximal portion, or this side, and then we look at the distal portion, this side, what's the difference between, say, let's get rid of all this text, what's the difference between this white tissue and this white tissue? What's the one up here and the one down here? Yes, one is a tendon, because you can see again connected to muscle to bone, and you can see that this patellar ligament is going to be connected to two different bones, and that's why you can see it's called a ligament, and why we're ignoring this one on this whole figure, but I draw your eyes to it because it's going to be relevant today. And so otherwise, ligament connects bone to bone. Big difference. You're seeing the difference right in front of you. All right. And so with that, you only have to know one example. You technically have way more synovial joints, but the only one I ask you to know is the knee joint, because we're all going to have knee problems at some point. And we're all sports fan. We all follow some sport. And you may hear the word, oh, they have an ACL torn or something. You never really know what those are. And that's my goal for this slide, is that you just have a general idea of where these slides are. So with that in mind, when we look at this now, to begin our journey into appendicular, the lower appendicular, we know what that large femur bone is called, right? Because we know it's going to take up the femoral region of your thigh. And then in your knee area, in the front, you have the patellar region, patellar with an R. And then now it's called after the patella bone. It's named after that. And so this little kneecap bone sits in front of the femur, and two paired bones, the tibia and the fibula. This is like having our radius and our ulna in the antebrachial region. You have the same thing going on down here. You have one long bone, which is comparable to the humerus, and then you have two paired bones that are going to connect to your phalanges eventually. And so you can see that in this this crural, sural region in your ankle, shin area, you'll have your tibia and your fibula. And again, that little kneecap bone sits right on the anterior side, nothing in the back. So with that, these ligaments are named according to the bone, so they're really easy. For example, tibial, fibular, collateral ligament. Typically, collateral means a result outside of the intended goal. That's why you hear, collateral damage like military operations. Like sometimes to destroy the enemy, you might hurt civilians. That's called collateral damage because it wasn't the intention, but it is what it happened. And so collateral is sort of like the side result of not your main objective. And so this is the same concept. Collateral ligaments connect on the outside. And so you can even see it tells you what side. Tibial is going to be connected to the tibia, to the femur, and the fibular collateral ligament connects the femur bone to the fibula. And so they're just named after that very fact. Collateral meaning on the outside. If you ever hear LCL, they're talking about the lateral collateral ligament. You hear MCL, they're talking about the medial collateral ligament. And this tibia sits medial. And so typically that's why this is our sort of our intro into our appendicular for the lower end, is that tibia is going to be medial, fibula is going to sit on the lateral side. Patella is going to sit in the middle, kind of floating between all those ligaments. We'll see a little bit more of that later. And finally, here's our patellar ligament, which is on the outside of the capsule too. Now, where it gets a little tricky is that if we grab the same joint and we flip it around, we can see the posterior portion. Again, let's orientate ourselves. The femur bone, the tibia, the fibula. This is lateral. Let's make a note to ourselves today. Lateral. Tibia is going to be medial. And so if you look at these ligaments, you can see they're labeled accordingly, the collateral ones. And then finally, there are some that go through the joint, meaning that they're going to go in a they're going to go right through it from the front to the back. They're going to originate somewhere here underneath this patella and make their way to the back of the actual joint itself. And so some of these ligaments are going to be named after that. And they're on this page here. You can see them, the cruciate ligaments. Like I said, cruciate means to cross over. And so if you look at the anterior one first, right now this bone, right off the bat, you are looking at it from the anterior aspect. Let's write that in here. Anterior. You're looking at it from the front. And what word in here should give me the clue that it's the front of that joint? Patellar. Patellar. Patella is going to be the front of your knee. And so this kneecap, if you remove that patella, you'll see two ligaments that make like an X. One runs above on the front. The other one runs behind it. And so logically, anterior cruciate ligament is going to be the first one that you would touch in case you were trying to feel these ligaments. It's the first one, more superficial, coming from the anterior aspect. And then the posterior one is going to be right behind it, crossed right behind it, underneath it. And so that's why they're called this. So cruciate means that it's going to run through the capsule, underneath, through the joint, and then collateral means outside the joint. That was the biggest difference, outside. You can even see them here again. Collaterals, cruciates, big difference. And with that, the next thing, you'll see our menisci. This is the better picture that I mentioned, is that you have two. You have one on the middle aspect and you have one on the lateral aspect. The medial one is going to be on the side of the tibia. So again, even going into lab next week, something that I hope that you kind of remember from today is that the tibias always, as you look at the inside of your knees, if you do this, you're touching your tibias. Remember, it's between or on top of your big toes. And so your tibia will always be medial on both limbs. So in this case, tibia is going to be your medial one, lateral is going to be your fibular one. Sometimes they're called fibular menisci. Actually, no, they're never called that. Never mind. I take it back. Menisci, again, will be that little piece of fiber cartilage that's just like the intervertebral disc, whose goal is to prevent wear and tear from the femur bone above, from grinding down on your poor tibia on your daily activities and exercise. What do you need to know as students here? You just need to understand the basic concept behind the word collateral, the basic concept behind the word cruciate, right? That they cross through. Collaterals do not. And then two simple concepts. Popliteal ligaments are going to be found where? Posterior or anterior? Posterior. So let's write that in there. You should know this just by second nature if you know your anatomical regions. And then any ligaments called patellar will be on the anterior side. All right, and do not forget about your menisci. And hold on to fibrocartilage. But otherwise, just knowing, just general simplicities out of this is good enough for me for this slide. There's enough on this exam. Just understand collateral. And tibial and fibula, by next week you'll know what those things are. Should you know them very well, which one's medial, which one's lateral. You'll be able to answer anything based off of that. So don't spend too much on this. Just understand these basic concepts around ligaments. And so now you understand what ACL tear is. It's an anterior cruciate ligament tear. And you don't need any of this general information that's here. It's here just for you for reading. All right. With that. So now as we look at dislocation is going to be different from fractures. And this is when we're going to come back to fractures. And I wanted you to see the difference between a knee dislocation and a knee fracture. And you can see a dislocation is going to be exactly what it sounds like, is that the bones get their locations get moved. And so you can even see our poor femur here is no longer sitting comfortably above the menisci, and so, or evenly between their matching menisci. And so these condyles, when they're looking for the condyles of the tibia bone, they always want to sit on top of their menisci. And so when that placement gets removed, we also refer to it, or dislocation can also be referred to as luxation. You may hear this. You'll rarely see this, but just know dislocation. That's good enough for me. And something that we addressed about our SAD scale on the previous lecture, is that we saw that if you are in the synarthrotic region, that means that you have no mobility. But at the same time, this guarantees that you are going to be the most stable. You're going to have a lot of stability because you don't really have a lot of movement. You're glued very closely together for simplicity's sake. And so synovial joints, they were the most diarthrotic, meaning that they are the most mobile joints. They are the most diarthrotic. They are classified functionally as diarthrosis because they move so much. But this in return causes low stability, or again, we call them synovials because they're very mobile, but they have the least stability because they are the most mobile on our SAD scale. And so they're the least stable. And so you can see that this is what this text here is meant to reflect. So this is something you should be able to understand after you've conquered the previous topics, that as you read this, synarthrosis on our SAD scale is going to be the strongest joint because it is the least mobile. And you can see all they're doing is regurgitating that information. Synarthrosis, strongest joints, have no movement, least mobility. Diarthrotic on our SAD scale is the most mobile. Therefore, they are going to be the weakest joint. So this is just a reflection on that very concept. Now, the word to describe a dislocation, its definition is based off of a previous vocab word on the first page of joints, range of motion. That was also abbreviated for you as ROM. And so range of motion is going to be whenever a joint or the motion goes beyond the range of motion, that is the definition of a dislocation, beyond the range of motion. And then some articulating surface was forced out. Because everybody does have a different range of motion depending on your conditioning, your genetics. And so some individuals might have more flexibility, like I'm sure a lot of you can't do this, right? Like this one all the way down here, all the way this way, right? So like this one that's a range of motion for most of you normies is like this, right? Like this. This is what you guys look like to me. And then so like this one's gonna be an example of low range of motion, but this is normal range of motion, and this is kind of overdoing it, right? And so range of motion is your functional range of motion. And obviously it's gonna be different. So a dislocation is only when things get popped out. All right? And so with that, anything else? That's really all I care for you to know for dislocation. And we're going to have to, unfortunately, revisit our fractures very momentarily. And so let's go back to the previous packet, which should look something like this. This is the old one. Let me use the nice one, the new one I made here. Oops. Oh, it's on the same packet, sorry. I combined these this semester and I forgot. So. Page 9, and I added page numbers too. Nice. I did, nice. All right. And so when we look at fractures now, fractures is going to be when the actual periosteum, crackers. And so when you look at, say, this, where was I here? Fracture repair, you can see that when the bone, the periosteum is broken, completely different steps, right? We're not talking about bone-to-bone interactions. We are seeing an actual fracture within the actual bone itself. And so there are 4 steps that you can summarize a fracture. First, a blood clot formation. That's what a hematoma is. It's a large blood clot. Maybe that word is missing there, blood clot. And it seals up, like it kind of fills up the space as a fluid. And we've all seen this process. You get a cut for a little bit, and then you see it getting all crusty. That's what a blood clot is. And so all the blood vessels do that for the bone to create some sort of texture that's going to get solid. And a quick little connective tissue question. What are the little tiny little cell fragments called that coagulate blood? Platelets. And so platelets are going to coagulate this and seal it up. We call that sealing. It's done by a hematoma. Next, you'll see that a callus formation happens, an internal one and an external one. If we look at our photos, you'll see that it's going to be made of 2 entirely different content material. And so you can see that our spongy bone, the internal one, where typically we always find spongy bone, try to remember that you only find compact bone on the outside of any of these figures that I'm having trouble finding for dramatic effect here. Okay, I'm just going to go ahead and switch up to the other one here. All right. Here. And so this photo here on the very center, we saw we had spongy bone, so it should make sense to us that if we fracture the outside, that the internal callus forms inside, external forms outside. If you can remember this, you can remember that spongy bone would be found on the inside, and on the outside, you do not care about the word spongy bone almost at all. And with that, back to where we were. Here. You can see that our spongy bone and the inner edges start to form, and that's what a callus is. The internal callus is just a spongy bone reforming. What's going to be building that spongy bone? What cells? Which cells? Osteoblasts, because blast build cells. And our external one, you're going to see a trend. What did we see in the fetal skeleton? What was used as a blueprint or a scaffold? Cartilage. And the external callus is unique because of that, because it has cartilage. So the chondrocytes come to the rescue during a fracture, and they do exactly what they do in a fetal skeleton, is that they serve as an initial blueprint. And so cartilage grows in that area, and then just like before, bone or osteo cells, at some point osteoblasts come in, take over, remove those cartilage cells, and lay out osteoid bone matrix, mineralize it, and then that's what happens at the external callus. That's only the real fundamental difference, is that an internal callus and an external one forms. The external one has cartilage, the internal one does not. And then on our third step, our bone comes in, or our bone cells come in to replace that cartilage. Here, let's highlight cartilage. It's going to be replaced by bone. And then this is where we get slightly complicated, is that when we look at our external calyx, That external calyx in the beginning, even though it doesn't demonstrate it here, keep in mind that it's going to grow very fragmented, and fragmented bone is spongy bone in this case. And so it makes what you know as spongy bone on the outside after the cartilage step, and then it turns that spongy bone into what we remember as compact bone. And remember that compact bone is always found on the outside. So that's the only little nuance here, is that the external calyx started with cartilage, number 1. Then it formed spongy bone, number 2. And then here at number 3, our compact bone formation is made at the external calyx. Yeah, those steps maybe need a little bit of revision, I think, but that's the general premise. And what's left over after remodeling, the scar of a fracture, is referred to as just this callus, this visual callus. The same thing that was formed out here, the external callus is what's visible, and it'll always be there, and it'll actually be a little stronger in that region. That's why sometimes they say that when you break a bone, it's a little harder to break it again in the same spot, depending on the type of fracture. And that could be because, again, this callus is more bone material, making it a little tougher to encounter or break in that particular region. Now, with that in mind, there are different fractures that are possible. These are only 3 that I'm showing you here because I think there might be the easier ones to grasp with the intro bone anatomy that we learn. And the easier one first is green stick fracture. This is when you have a slight fracture on one side, and out of this scan here, what bone is this one? What was our biggest clue? What can we use here? Thumb side, good. Thumb side, radius side. So our partner, what's our clue for ulna? Pinky side. And so with that, let's keep going. And so this fracture, when it occurs, sometimes you can see a bending onto the other side. And so when you see some sort of, destabilization of the overall shaft because it pushed in on one side and pushed out on the other, that's called a green stick fracture, where one side gets broken and the other side gets bent. And this typically happens in children, and we should have a general understanding why. Recollect that this overall process we talked about, these 3 steps, is a transition from infancy, or actually let's do these 2 steps. This is what it looks like growing up. And when does it look like this? Adulthood, or at the end of puberty, where this is no longer cartilage allowing for growth. And so the bones are a little weaker at some points because of this, and there are fractures named after that. And so remember that just children's bones have a lot more cartilage than adults do. And so you can see why they might be easier to break versus others. And in general, this will always be a fact for children versus adults, is that children do break their bones a little bit easier than adults. And for this very fact, long bones have yet to fully ossify, to fully turn into bone material. Then we have transverse fractures, which are pretty easy. That's why I picked this one, because if you have a transverse fracture, it looks exactly as if you're making a transverse section on anything, like this. It just splits it, and these are generally pretty easy to fix because you just have to line them up together and then secure them, bolt them. It just really depends on where, the severity, and other tissue damage on the side, and bone fragments might break out, and you got to get rid of those. So it just really depends on what's going on. But for the most part, transverse tend to be the simplest because you just line them up and they heal nice, pretty evenly well again. And so transverse fractures, easy to identify. Last, spiral fractures. These are going to be a little tougher, but this is like if you grabbed a towel or a shirt or anything, and that's like if you do this happens, this typically is in car accidents that you see it, like a leg gets trapped, and in the flipping and in the leg gets trapped, and you see that the leg undergoes this torsion, like this. And this is a torsion fracture or a spiral fracture and this is the most damaging because it's like, again, it's like actually grabbing a solid material and just really destroying it like this. So those tend to be the most difficult to heal, and they're produced by that twisting stress. And so for here, just no transverse traction. Look at clues, right, along a transverse axis. I'm going to be nice here. Transverse plane. You just have to be able to match it with the clue. Transverse plane, transverse fracture, spiral, twisting is our clue. And green stick, one side broken, one side bent. Those are the clues I need you to know for these three. And these are just three examples. These are fun to look up after probably when you have some free time, and I would recommend it. All right. So I believe we finished with these slides, if there's not anything left. Just our motions. And the motions don't worry about it in your lecture exam. Keep these and take these into when we learn muscles, and I'm going to add it to the lab exam. It's already part of the lab exam, actually. Yeah, so this portion, keep it in your notes. We're going to need it again when we get to the muscles in our lab. And I think that I'll test you for it in the lab this semester. Yeah, all right, so let's move on from here to our next packet for the sake of time. All right. Now, what you are learning today is muscles and excitation, contraction, cycling. Muscles is pretty easy. We all have a general idea of what the muscular system is. We all see it on top of the muscles in our lab. And so this word itself is a fancy word to mean the activation of a muscle. Do not let this word intimidate you. This is the first thing you should learn to get it out of the way. Excitation means exactly what you think, to receive some sort of stimulus, and let's write that. And contraction is to, like an accordion, to squeeze. And we're going to keep it simple for now, just squeeze. And we'll clean this definition as we move along. Squeeze. And as we look at these pictures, when you look at a generic muscle, don't flip slides just yet. These muscles that you see on our models all over the lab, you may see that they run in fibers, and every single one of them is made like a bunch of little strings, thousands and maybe into the millions of them, completely compressed together like a bunch of straws together. And each segment, like this is a famous muscle that we all, see some people flexing at the beach, right? The biceps brachii. And so this is the biceps, the one that you flex. And so just this one is called the biceps. And if you zoom in on it, it has to always, you'll see that it's connected to the brain. The spinal nerves travel through the vertebral column, exit the vertebral column, travel somewhere around that clavicle and that scapula, and that nerve goes down through the head of the humerus, travels through the deltoid muscle, and it still travels, and then it finally connects to this muscle. And when you want to actually do what this man is doing on the screen, bend your elbow, just bend it, he has to send that signal to that. That's exactly what this means. You are sending a signal from your brain, the stimulus, for the muscle to move or squeeze, which is what a contraction is. A contraction is the actual action a muscle does. Action a muscle does. Yeah, that could have probably been expressed better, but actions that a muscle does. And what they generally do is squeeze. And before we get into that kind of like motion, because that's more relevant for lab, you want to just understand how it receives its signal and how it's actually going to carry out the contraction. So you are seeing your entire chapter summed up in this image right here. My goal is to have you understand everything going all the way to this very region up here. This is what physio really stresses on. So this is something you're going to encounter when you get to physio. But everything we discuss in anatomy is up. So first thing, we begin with neurocontrol. You can see that this is a general brain cell, a neuron, with its axon connecting to what is a muscle, the boundary of a muscle. And this is more another drawing of the same representation, that you can see that the axon terminal comes to the very end, and it's going to connect to a muscle cell. And if we zoom out even more, this is an even better illustration to hold on to. This is a muscle fiber. And remember that the word muscle fiber is interchangeable with what word? Cell. And so when you look at this proportion here, the word muscle fiber is the same thing as a cell. What are some organelles that cells have? Mitochondria. We're even seeing these right here. That's what these little roly-poly looking ones are. Mitochondria. What's the membrane of a cell called? Plasma membrane or cell membrane. What is the headquarters, the thinking, the DNA storage site? Nucleus. What is the protein making little tiny ribosomes? Those ribosomes can be attached to what sheets? The rough endoplasmic reticulum. And what's the other one that's not rough called? What's made in the smooth? Lipids and carbohydrates. And what's the last little organelle we're missing there that transports? Golgi apparatus, or Golgi body. And so this cell has all those properties. You know absolutely everything about this cell. It has a plasma membrane. The only unique things that you have to add is that many nuclei, multinucleated. Other things, it has mitochondria. What does mitochondria do for a cell? ATP. So you can see that all our little images stress it because our muscle cells need a lot of energy. And so with that, we move on. And so some terms around this is that we call this connection of a nerve cell to a muscle a neuromuscular junction. Or for short, I just call them NMJs. But it's called neuromuscular junction. And the definition is here. A location of where a motor neuron controls a skeletal muscle fiber. This is such a key thing. You only really have, in this class, two types of neurons, motor and signals of motor. Do they go away from the brain or towards the brain? Away from the brain. Think about it, right? The brain is telling the bicep to move, so it's going away and connecting to the muscle to move it. So as you look at me send this signal, it goes from the brain, sending it to this muscle so that I can flex it or extend it. And so that's going away, motor. Everything going away will always mean motor. Because think about your body. Your body is not that complex. When you think about the brain has two things. It wants to sense the world, and then it wants you to move. It doesn't really do much. Any of the complex thing that we discussed in this class so far is that it wants to sense the world, and it wants to be able to move your body in response to the world. Motor, it means to move your muscles. Your will, your conscious free will, like, you might have taken philosophies, is free will real? And you know how I know it's real? Because you can do this. And there's no purpose for it, but I can do it. Free will is coming, your ability to decide to do something about your dreams, your goals. You have to move your muscles to do it. Even in this class, you have to grab your textbooks, you have to put them somewhere, you have to open pages, you have to move muscles. Your muscles are your free will. And so motor goes away. And sensation is the opposite of acting on the will, right? Acting, when your senses are there, victims of the circumstances. Like right now, you have no choice but to listen to me because I'm talking, right? Whether you want to listen to me or not, you're hearing me because your body is an antenna. It's receiving light. It's receiving sound. It's receiving textures. So it's just like this, like it's easier to understand what I'm saying if you just close your eyes, because the sense of vision blinds the rest of them. And as you close your eyes, you can hear me talking, you can hear the sound, you can even tell how hard this material is versus like this. And so you can see which one's harder. And so again, you are always sensing things. And the reason this is relevant is because motor neurons are the only things making neuromuscular junctions. The only opposite of motor is sensory. And that's going into the brain. Into the brain. Brain. And this is not a subject for today. All right, but it will be for nervous. And if you understand these two basic concepts, nervous will be easier because you would be so surprised not being introduced to that early enough makes the whole nervous chapter hard. So with that, we move on. And so this axon ends. Actually, I have better pictures of this. Let's actually just flip pages now. All right. And so something I could on here in chapter 9, muscles. But otherwise, when you look at muscles, something to keep in mind is that the star of the show for today and our exam is going to be skeletal muscle, but they all operate essentially the same. Skeletal muscle was particularly unique because it was voluntary. 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 And the word contraction means, again, to squeeze. It is the action of it. I'll give you more illustrations of that in the lab. And when we get to the 3 types, you know them all: skeletal, cardiac, smooth. Let's start off with the ones that we don't care about too much for this chapter. Cardiac muscle tissue, we already know everything. It's involuntary, found only in the heart. We know what the heart does. It circulates blood through the blood vessels. Smooth muscle, we know it's found in the hollow organs or digestive tract. One that I want you to start being aware of is that it's found in the arteries. And then it is also involuntary. We do not control it. In the integumentary system, we find them as well. What were they called in the integument system? Anybody remember? Starts with an A and the second word P. Think of the word erect in a way. Rhymes with it. Erector pili. Who said that one? There we go. And then so rector pili. And so as we look at this now, when you look at this, now we go back to our skeletal muscles, and that's really the topic of today. And this is what we want to make sure we know our functions. You should know most of them. Produce movement, maintain posture, body position. These are going to be easy to recognize. It's all multiple choice. Support soft tissues, a little bit tougher, but the easiest way to think about this is that they do shield internal organs. The skeleton is completely covered by them. It guards entrances. You have sphincters, for example, the anal sphincter prevent, when you're in a classroom and you don't want to miss that last bit of notes, there's only 10 minutes left and you're like, I can hold it. The reason you can hold it is because you have a muscle guarding the exit. And so that's referred to as a sphincter. Even right now, me talking, this is a sphincter. Even your mouth is a sphincter because it regulates entry into an entryway, an opening. And so that's what a sphincter is, or the overall function. Guard body entrances and exits. Even today, or in lab this week, you learned your orbital, right? And there's a little sensitive little eye sitting in there. Your eye is a sphincter too because they can even pucker like your lips. And then you even do it, right? Someone's going to poke your eye and it can protect your eye. So it guards that entrance as well. Maintains body temperature. All those people who have their lives together and run in the morning, they're capable. They're running in shorts and tank tops, and it's freezing outside because they generate heat. And then lastly, they store nutrients. And when we say nutrients, one major store that they have is that they store proteins. They metabolize them very well. This is why you see everybody drinking protein drinks all the time after workouts. And then they also are the greatest store of glycogen in mammals, in any organism, really, but glycogen. Anybody remember what glycogen was? Sugar. They're the long branches of sugar. Glycogen. And so that sums up this page. Now, here, I used to maybe try to get more complex here. I'm going to keep it very simple. Remember how every tissue has stem cells? A muscle cell's stem cells are called myoblasts. That's all I kind of care for you to really know. And then myoblasts, remember how we had osteoprogenitor cells that can turn into osteoblasts and osteocytes or osteoclasts. These myoblasts can turn into myosatellites. If there's enough time, I will come back to anything more I want to add here. But this is kind of the segment I already foresee I'm going to end up cutting off a little bit. So if you hear these words, stem cells, right? The stem cells for building and repair. And if anything, I'll talk about it next Tuesday, but I won't put it in the exam. But I want you just to hear these words and know the basic function of these words, stem cells. And so looking at our pictures, you can see that these myoblasts will eventually turn, or maybe myosatellite cells, and then when they need to repair a major cell, then they fuse at the edge, and then they can fuse with it, but more on this if there's time for it. So this is something I foresee I'm going to have to cut out for the sake of time. All right, let's move on. And so now something that is the most difficult thing to grasp about muscle cells is that I keep telling you, I'm insulting you a little bit and making, because it is hard, but I keep saying, oh, cell, yeah, nucleus, it has the Golgi body, the rough endoplasmic reticulum with all the ribosomes and the smooth one right next to it, and then it has all those lysosome, peroxisomes, and I might be missing other organelles, but otherwise it's the same exact thing that you're seeing up here, but the big difference is that these cells are huge by comparison. This is what a cell would look like, a regular cell that we've explored, and you can even see the skeletal muscles are unique because they have many nuclei, because they need them. These cells, if I drew another cell next to this one, another regular, like a little squamous cell, a little columnar cell compared to this muscle cell is about this big. Oh, let me draw it in. Sorry. Like this. This is what a columnar cell looks like by comparison to a muscle cell. This is what a cuboidal cell looks like by comparison. A squamous cell probably. A fibroblast, a chondrocyte, an osteocyte. So you can see all these cells are that big, and a skeletal muscle is it's not even finished showing you on this picture. And so they're huge by comparison. They can be up to 30 centimeters in length. That's how long these cells can span to. And they make sense, right? Because if you think about the fibers, like this bicep, like you can see that it starts from here and goes here, right? And so those fibers have to reach from point A to point B. This is obviously a small muscle compared to one that we're going to explore on our back that goes from the skull all the way down to that sacral bone. And then so obviously not 30 centimeters, but they can be. And so you can imagine that they're incredibly, incredibly long. And therefore they need more brains. They need more nuclei in them. And so that's why they have more nuclei. And these words around it, sarcomere is going to be the main functional part of a muscle cell. Sarcomere, we're going to see that in a second. And everything is named after that. You already know it. The cytoplasm, all you do is you just change it to sarcoplasm on a muscle cell. Same exact word. And then plasma membrane, sarcolemma. That's the different word, sarcolemma. But it means the same exact thing as a plasma membrane. I think I just actually misspoke, sorry. Sarcoplasm, cytoplasm is the space inside the cell. Sarcolemma is the plasma membrane, the cell membrane of a muscle cell. And remember, one thing that characterizes muscle cells is their many nuclei. And this again can be attributed to their length. Now, this slide here. As we're going into the organization of a cell, we have to establish where we're at. And so, what we're looking at here, this first picture, you can see that I'm starting small here. This is a muscle fiber or a muscle cell or a muscle fiber. And I'm highlighting this in red up here, muscle fiber, because this is the hardest part about all of this, is how muscle actually organizes itself, but you don't want to forget this figure. This is what a muscle fiber looks like. This is what a muscle fiber looks like. This is what a muscle cell looks like. The same thing. And so, this is what we're looking at now if we were looking straight at it, right? If we grab one of these cylinders, we turn and we're looking straight at it, you can see these words inside of a cell. It doesn't really have much free-floating cytoplasm like in the other cells we saw. Instead, what we see inside of these cells are a bunch of actual little protein machines called myofibrils. That's really what skeletal muscle cells are made out of, is that they're made of fibers, muscle. And these are even called muscle or tiny muscle fibers because of this. And that's what makes this so annoyingly confusing, because this whole thing is a muscle cell, therefore it's also a muscle fiber. But this in Latin literally means muscle, tiny fibers. And so inside of the muscle fiber, you have myofibrils. That's usually the best way to think about it. Inside of muscle cell, aka fiber, we have myofibrils. And I promise in your studying, please start with those definitions. Do not move on until you get those two down. It is incredibly frustrating how confusing that can be if you start off just trying to get it all too fast. And so this myofibril here, these sit like cylinders right on top of each other, like straws inside of a container, but it's just sideways. The sarcoplasm is the cytoplasm in between all of this space. Sarcoplasm, cytoplasm, same thing. All right. And as we move on, and we can see that these are the same structures. The myofibrils are all those little cylinders. They're all sitting within the muscle fiber. You can see the mitochondria, bless you. You can see the nuclei on the edges. Now we're zooming out now. We're getting larger. Now we can see this is our muscle fiber. Oh, sorry. Pardon. 1 second before we move on. All right. So if we take out one muscle fiber, okay, sorry. Okay, now we're back to, okay, here we go. And so this is one muscle fiber, and this is where I tend to lose everybody. This is why I'm going as slow as possible. Here, muscle fiber, this is just one of them. Now, if you look at the higher level of organization, pay attention to this one, and then I want you to know all these segments. Each and every single one of these is a muscle cell or a muscle fiber. I'm just going to call it a muscle cell from now on to make life easier. And that's what's highlighted here. So you're seeing 1, 2, 3, 4, 5, 6, 7, 8 of these things piled on top of each other. And that's what you're staring at when you look at this. Each one of these. So there are approximately 8, if I just counted that right, 8 of these muscle cells within there. And each one of those muscle cells, that little group, is going to be completely surrounded by a tissue called perimysium. Perimysium. And if you look at just every individual muscle fiber, each one is going to have a connective tissue inside of it, separating them, called endomysium. And so, with our picture, endomysium is everything that's pink in between. Let's highlight them, right? That might make it easier. And it's connective tissue, probably areolar. And then that's going to be endomysium. And our perimysium is the one that envelops the entire region. Perimysium. And each muscle fiber has an endomysium. All right. And so it gets even crazier, because then this group is then further grouped into what we're looking at the very top up here. And so let's organize what we're seeing. Each one of these was a muscle fiber. Let's actually designate values. I think that'll be easy. I never tried that. Let's try that here. Let's pick a different number here. Here, color. 1, 2, 3, 4, 5, 6, 7, 8. And I'm going to call it, I'm just going to call all of those together, the 8. Okay? Because when we look up here, each one of these is that same 8 bundle repeated all over again. And there's obviously more in some of these photos, so I'm afraid of doing that. But this is the designation that I want you to understand, is that all of these together are grouped, and now you're just seeing one of these in just one of these circles. And then it repeats itself. You ever seen those dolls? The ones that you open it and there's another doll inside, and you keep doing it, you keep doing it, you keep doing it? That's what's going on here. You're just seeing structures within a structure, within a structure, within a structure. And that's why this part is one of the toughest parts is the organization of how muscle organizes itself. But start small, one cell, then a few cells grouped by perimysium, and let this tissue help you with the organization. Because if you look at each one of them, you'll see that every single one of these circles has that same texture around it. That same texture is your perimysium. Perimysium. So let's finish our highlights. That'll hopefully make it easier to translate this bottom image to the one on top. It's kind of already like that in color, but I know some of you don't have color. So I'm making this for the video so you can highlight this at home. All right. And that's perimysium once more, same color. And then our endomysium, again, is always going to fall in between all of those muscle fibers, all the tissue within there for the endomysium. And you can even see that's why it's pointed there. And the reason the perimysium is relevant is because it contains blood vessels. Contains blood vessels. And then lastly, the last final wrap is going to be epimysium. And that's exactly when you look at the very surface of muscle, the first thing that you see when you look at each muscle is that the first thing you're looking at is the epimysium. And that's going to be a connective tissue that wraps around it, that keeps it all contained and together. Each one has its own epimysium. Every huge muscle looks like this when you zoom in on it, and it just keeps going down to the muscle cell. All right. And so this becomes, make sure that you know the order of them. You have one epimysium around an entire muscle. You have a perimysium surrounding a bundle of muscles. A bundle of muscles like this altogether is called a muscle fascicle. Muscle fascicle. That word is up here for you as well. Muscle fascicle. So this whole circle is a muscle fascicle wrapped by a perimysium. And you can see the same thing is true up here. The muscle fascicle or the bundle is wrapped by a perimysium. So we're just zooming in. We're zooming in to see that image right below. And so this will require a little just evaluation until you finally get them. All right. With that, we can move on and pay attention to what the study guide asks for these very portions, and you'll be fine. Next, we have our tendon. And a tendon is going to be all of these tissues combining together. If you look at these cylinders, right, no matter what level we started at, whether it's endomysium, whether it's perimysium, or whether it's the whole entire epimysium, they're all going to eventually connect and combine together. They're all going to eventually go and meet here and form what you know as your tendon. Your tendon. And so the tendon is really all of these mysiums, these tissues, the epimysium, the perimysium, and the endomysium all coming together at meeting the bone, connecting to it, and that's what a tendon is. And that's what a tendon is. You'll also see the word aponeurosis, and a tendon is like a string connection, like a rope. I'm going to put rope instead, like a rope connection, rope to bone, so to speak. Rope to bone. And then I'm putting it as an expression because it's not the best way, but it's the easiest way to think about it. Rope to bone. And then aponeurosis is like a sheet, Like think about a blanket on top of this table and it's glued onto the table. A sheet on bone. Aponeurosis. And again, think of like a blanket glued to this table and if you start ripping it off, it's like, that's exactly what aponeurosis is. It's like a sheet that's exactly like a tendon, but it's laid out in a different way. And anything else? And don't worry about the actual contraction cycle like this. Ignore this for now, for today, right now. And that's it. That's really it, is understanding this figure, do the reading behind these terms to help you understand what we just discussed on the organization of this segment. So with that, now we're going to zoom in on where all the action is on the muscle cell. We're looking at the muscle cell again, and we are seeing all these little myofibrils. And inside, those are those little straws that I mentioned, those myofibrils, in Latin meaning muscle fibers, again. But otherwise, these myofibrils sit inside of the muscle cell. And when you look closely, you can see that they actually have these unique little structures on them. And that's what we're seeing when we zoom in on this. That when we grab one of these myofibrils, and this is what we be seeing. And the cool thing is you already technically know what a lot of this is. Do you see any ribosomes on this? No, we do not. So this, and what contain the ribosomes in a regular cell? The endoplasmic reticulum, the ER. This is the ER of a muscle cell. It's just in a different style, different look, and it has these yellow bands that we're seeing here, or these bands that run in the middle between them. We'll talk about what they're for in a second. And you can see that's continuous for all of them. And this is going to have a big role in the muscles because this is where they store calcium. That structure, which I'm not seeing it labeled here for some reason. It's probably on the next one. And so when you look at, I guess it's on the next one. Should be here. What's on the next slide? Oh, next one. And so the only thing we got to grab from here, each one of these cylinders is a myofibril. Those striations you saw in lab, those pinstripes, these that you saw that you were asked for, they're actually a result of the arrangement of these myofibrils. The way they arrange themselves, and they all contain their endoplasmic reticulum right on top of those structures, make up that unique pattern that was those striations in your muscle cells. If we zoom in on them, right, and we remove our endoplasmic reticulum out of this, remove these two tubules, we would see these little structures inside. These myofibrils, these cylinders, are made out of proteins. One is called thick filament, the other one is called thin filament. And so as you look at this portion, thin filament, thick filament, they are made out of different things. Thin filament is made out of actin. Thick filament is made out of myosin. Thin filament, actin, thick filament, myosin. Sorry, I think I said the opposite just now. And then these two are the only myofilaments, or all the myofilaments we find here. And this word is just as frustrating because this means just thinner fiber in Latin. And myo will always mean muscle. Just like osteo means bone, chondro means cartilage, myo always means muscle. And to be more exact, it actually means mouse. You'll actually see a lot of things that are related to mouse are called myo. And that's because the first time that people were describing muscles, when you rotate them, right, like you rotate, it looks like mice is crawling underneath the skin. That's why they called it that. And so myo is actually mouse in Latin, but in anatomy, it's referring to muscles. Remember that. And so these are incredibly thin protein fibers made of thick and thin. And you can see that these microfilaments are visible as purple on my screen and pink for thin filaments. And so we move on from there. Surrounding that thick and thin filaments is going to be the sarcolemma. And the sarcolemma, thank goodness we know what it is. It's going to be all these myofibrils are surrounded by a plasma membrane, a sarcolemma. And thank goodness we already know what that is. That's all I need you to know right now. And this next term I want you to get is that this word T-tubule. This word T-tubule, or this term part in this structure, is that it gives a signal to the structures next door the endoplasmic reticulum next door, it gives a signal to it. A signal. And it gives it the signal to release calcium. To release calcium. And we call the endoplasmic reticulum inside of a cell, we call it sarcoplasmic reticulum. Endoplasmic, sarcoplasmic reticulum. And again, the major fact of this that I want you to know about the SR is that it stores calcium ions. It stores calcium ions. And that's what this picture is trying to show you there. The inside of that sarcoplasmic reticulum, and then it's showing you the outside. This is the cytosol of the cytoplasm of the cell, or the sarcoplasm. And so this is the fluid around the muscle cell, and this is just inside those blue sacs. And that's storing a bunch of calcium. That's what all of these dots are, and that's going to be relevant in a second. These three structures, you'll notice they make a 3. This is called a triad because of that. And this triad is made out of two pieces of sarcoplasmic reticulum called terminal cisternae. You have 2 of these and 1 of these in the middle. And that's why it's called the triad, to make 3. And so you can see the 3 bands here. We have 1 in the center, T-tubule. We have 2 and 2 for terminal cisternae. But all 3 are a triad. And it repeats itself over and over and over again. And you can even see another one adjacent or next door. And so this T-tubule is necessary to release calcium from the terminal cisternae. And the last thing, what stores the calcium inside is a protein floating around, a protein embedded into the membrane of that sarcoplasmic reticulum. It has its own membrane, and it has proteins embedded into that membrane, and they're called calsequestrin, to mean sequester. Does anybody know what sequester means? To gather. So this is like saying calcium gathering protein. And it's a protein that binds calcium. It gathers protein. And we will understand this reading at the end, by the end of this talk. And so we will move on to finish up. And this is the last thing to mention. A signal must go through this T-tubule to release calcium from those terminal cisternae. And that's what this picture is showing. A signal must go through the T-tubule for the sarcoplasmic reticulum to release calcium. So this reading will make a little more, should make sense by the end of this section. And so let's move on to the relevance of this calcium, and to finish up what I believe is what we're going to get up to today. So underneath this sarcoplasmic reticulum, you can see there's position of M line. You can see it underneath there. And so if we get rid of that endoplasmic reticulum, we see these thin filaments, our microfilaments underneath, our myofilaments. And so a sarcomere has particular segments to it. It has a Z line, an I band, A band, M line, H band. You must be able to know the anatomy of it on a sarcomere. And a sarcomere, first one's Z lines. And even draw this Z line because this should be drawn here. Z line. This is one sarcomere. A sarcomere is from one Z line, from one Z line to the other Z line. And it repeats itself over and over again. Repeats itself over. Here's one. Here's the second one. And it just does it through the entire length of the whole muscle. And every individual unit is called the sarcomere. The boundaries of that unit are the Z line. You can use the Z to remind you of like zigzag lines, right? Like this, zigzag. And you can see zigzags. That's why they're called that. They're making kind of like Z's, and Z lines. Next, you'll notice that between two sarcomeres, because here's a sarcomere, here's the other sarcomere, that this part doesn't belong to that sarcomere at all. It doesn't belong. This part belongs to this whole thing. But still, this whole portion's unique because it only has thin filament. And so we call that portion with only thin filament the I band. And the part that only possesses thick filament is going to be called the A band. I always kind of remember artificial intelligence, AI, to remember the order, and I look at the thick filament and I just start there, A band, AI. And then the thin filament is going to be the only portion, thin filament. Next, that this center line, this thick filament, the middle of the sarcomere, I always know I'm looking at a sarcomere because it always has a midline called the M line, which is shown on this picture here and here. I'm sorry, I'm not seeing it labeled anywhere on, oh here, M line, right next door, M line. And it's the same one on this one, M line, the middle of the sarcomere. The A band was unique because it only cares, I'm gonna put only cares about thick filament. The I band only cares about thin filament. And now, the only thing to add a complication to those two concepts is that A band, no matter how this is shaped, no matter if the muscles squeeze together or not, A band is A band because it is only the boundaries between that thick filament and that's it. And that never changes. What does change is that eventually you're going to see that there's a space here where the thin filament is going to meet the other half, its other side of the M line thin filament, the matching side. And that's really what a muscle contraction at the end of the day is that if we look at just the thin filaments going towards that M line, that's all a contraction is, like an accordion. They squeeze towards that M line, and you can see that they did that very thing on the picture adjacent. That's what the arrows represent. And then this H band got smaller. And so this H band, it starts off a little larger when it's relaxed. The second you want to squeeze that muscle cell together, then those thin filaments travel, all of them travel towards the M line from each side, going towards that M line. And so this H band decreases inside. Everything that we're talking about right now is referred to as the sliding filament theory. This is just the way to describe the way a muscle contracts. Because if you look at the overall muscles, without making it so confusing, without M this, AI this, and all this, if you just look at these two pictures, you can see that this is the same thing, the same amount of sarcomeres squeezed together. And that's what muscles do, like an accordion, they do this. They get smaller. But there's just millions of fibers doing this all at the same, trillions of fibers doing this at the same time. And so they do so because the thin filaments from each side of the sarcomere will go to the M line, making each sarcomere smaller. So if you look at our picture, starting here at the center, you can see these thin filaments have to move to the center. They do so, and that happened. Our H band got smaller, and you can see that happens to all of them. And so they all do it, the whole entire muscle gets smaller. So what a contraction essentially is, that right now as you look at my arm here, this is, all of them are like stretched as much as they can go. And then the second I do this, all they're doing is that both muscles, the muscles from point A to point B, are all squeezing like this. And that's why when this squeezes, it pulls from it pulls these two bones together, the ulna and the radius, wherever it's attached. That's point A and point B. And then when it squeezes, it moves point A to point B, and then it causes the bending around that trochlear notch because the muscles here are squeezing together, letting the two bones fold on that trochlear notch going over that trochlea. And I hope that makes sense since we've been talking about appendicular. And so make sure that you can describe the sliding filament theory using these terms. And that's what this is really here for. Thin filaments are made out of what we refer to as actin Myosin is to make up thick filaments. And then thin filaments has tiny little active sites in this little segment here that we're seeing. Little active sites. And those little active sites are covered by another protein. And those protein that covers those little active sites is going to be referred to as tropomyosin. Covers active sites. And then you're going to have what I'm going to call, let's say, I'm going to make it easier. I'm not going to use that word. I'm going to call it the gatekeeper. I'm going to put the lock and key. Gatekeeper. Troponin. And without really using these words, essentially what thin filament needs to do is that it needs to interact with thick filament for it to do the sliding filament theory in order for these filaments to go this. These little heads need to interact with the thin filament. And so when you look at the thick filament, you can see it has the little nubs made of myosin, and it has a head and a tail. And they just repeat themselves over and over. And that's really all you need to know here. Head and tail. Head and tail. And they are always going to be looking for these active sites, which are going to be protected by tropomyosin. They are covered. And this troponin is going to be responsible for removing it when necessary, and that's why I call it the gatekeeper on my notes. Do not worry for our exam for these two at all. And let's flip our page here to finish up this last concept. And so to go from a resting sarcomere to having a contraction where those thin filaments go towards that M line, as we progress into this segment here, you can see that this is nothing but how they interact together. And the key player is calcium. Where in the muscle cell is calcium stored? In the sarcoplasmic reticulum. This is where that calcium comes from, and this is the key. The key is calcium. When we look at the thin filament, I said that this was the gatekeeper. Troponin binds to calcium, and when it does so, we can see on our corresponding image next door that calcium bound to troponin, the gatekeeper, and then tropomyosin got out of the way. And now all of our little active sites are exposed onto this little area. And these myosin heads can now bind to it. And then the cool thing is now that we see ADP and P, this means adenine diphosphate. If we add one more phosphate to this and turn it into triphosphate, what does this change into? ATP. And what is an ATP molecule? An energy molecule. And so you can see that your muscle cells require ATP, or the myosin head requires ATP. The thin filament requires calcium. And so when calcium is present, it allows for the active sites to be exposed. The ATP will be there to serve as the energy that it's going to need to bind to the active site. Once that active site is bound, we call that cross-bridge formation. This is technically a vocab word, cross-bridge formation. And it's on everything I'm saying right now is written on the next page for you and bolded for you, so just in case. But otherwise, cross-bridge formation. And now we have our head bound to our thin filament, and then what occurs is a myosin head pivot. And then you can see that the energy molecule gets used up, and then our thin filament moves towards our M line. Towards the M line. And then the next thing that happens is that was only one pivot, and we need that thin filament to go through several myosin heads. This has to go from here all the way here, and we just saw one pivot. So the rest of these steps is really just a reset of the simple movement that we just saw, where we need a reload energy. So you can see ATP comes in to reload. We call this like when you have a gun and you, they say when you hear the movies that they go, ch, that's called cocking a gun. Same concept. When we say ATP goes there, it recocks the head, and then it pretty much means to reset it or put it in a position ready to do it all over again, to be able to shoot that bullet, so to speak. But otherwise, it's ready to activate again. And so calcium is still bound. As long as we have calcium, these active sites will be exposed. ATP replaces the one that was used up, and then we see it is used up, and then we have another pivot. But finally, when the calcium leaves, what do we expect to happen to those active sites? They get covered right up, and then nothing happens. The contraction is done, essentially. And so we've reached pretty much that. We have a contracted sarcomere when we finally have the thin filament making its way all the way as close as possible to the M line, right? And they don't always need to go all the way down to that M line if you really think about it. If I'm holding this can, it's not that heavy, and to do this, I don't need all my myosin heads to go through the whole way. But if I'm, lifting heavy weight and it's heavy and I really want to make it all the way to this, then I have to, rely on a lot of energy and have to have sufficient calcium, right? And so there's a minute left, and this part is something that I wanted to add this semester. So this is the last little bit I'm going to record for you, and it'll be up by 5 p.m. tomorrow, or midnight tonight, or 5 p.m. tomorrow. And I'm going to make this part, these last 3 pages, the extra credit of the upcoming exam. So if you watch that video over the weekend, it'll be extra credit. If you don't understand it, I'm going to bring it back during Nervous anyway. So otherwise, it'll be the extra credit if you get it a head start on Nervous, because that's what this is.